Bacteria are classified according to a number of things. They can be classified according to their pathogenicity, to non-pathogenic and pathogenic. Non-pathogenic means the bacteria that do not cause any disease in normal situations as long as the body is healthy. Like, for example, the normal flora that exist in our intestine. They help in digestion and produce biologically important molecules like vitamin B and vitamin K. Staphylococcus bacteria exists normally on our skin and maintain its acidic pH that protects it. In normal situations, these bacteria are quiet, but if your body's immunity drops or if they somehow translocated from one location to another, for example, if you have a skin wound and these Staphylococcus bacteria penetrated to the blood, they start causing problems. Pathogenic bacteria, on the other hand, means the ones that cause infections and can lead to severe illnesses. And these are the ones we are targeting when we are using antibiotics. Bacteria can be classified according to its shape as well. Cocci means the bacteria that have rounded shapes, while bacilli means the one that appears like rods. According to growth requirements, the majority of bacteria need oxygen to grow, and in this case they are called aerobic, while others grow better in the absence of oxygen. Some of them live in our intestine and our mouth. They are called anaerobic. Another important classification that we will be using mainly during this course is according to Gram-Stein. We have seen that bacteria have cell walls. This wall consists of protein and carbohydrate. It's called peptidoglycan. Some bacteria have thick walls, while others have thinner walls. A scientist named Christian Gram once added crystal violet solution that stains bacteria to the violet color. He then added many reagents. Iodine to stabilize this stain. He added alcohol, and then he added another stain called safranin, which is pink. What did he find when he looked under the microscope after that? Some bacteria remained violet, while others were pink. But how is this explained? Bacteria that has thicker walls retain the very first stain it was exposed to in their cell walls. They were not affected by any other added reagent, while bacteria that had thinner walls were actually surrounded by extra lipopolysaccharide layer to protect it. When Gram added alcohol, it dissolved this layer. And when safranin was added, their cell walls were ready to be stained again, and they appeared pink under the microscope. That's why we can distinguish between gram-positive, gram-negative, and atypical bacteria. Gram-positive are the ones with thick cell walls that stain violet. Gram-negative are the ones with thinner walls and outer envelope of lipopolysaccharide that stained pink. Atypical bacteria were the ones that weren't stained neither violet nor pink. They either had no cell walls at all, like Mycoplasma. They may have had cell walls, but of different composition, like Mycobacteria and Chlamydia. Or they were difficult to be grown in normal laboratory settings, like Legionella.